Ece Akhan. I'm working as Biobank Quality Manager in Hacettepe University Rare Disease Biobank. I am also a member of BBMRI National Node. Together with my colleagues from different biobanks, we have developed this video tutorial specifically for rare disease researchers and developers. In this video tutorial, we started with explaining the challenges of rare disease and how biobanks find solutions to these problems. We presented you with the information about biobanks organization. We also guided you about ethical, legal, and social issues in biobanks. We were explaining how quality management systems works in biobanks and address the sample data management. In final part, we finished by highlighting the importance of biobank sustainability.
There are more than 7,000 rare diseases in the world. Unfortunately, only about 5% of them can be treated with current drugs. The reason of this inadequacy is because rare diseases are hard to diagnose. Still, there is lack of enough information and experience about rare diseases. Due to limited patient number and wide geographical distribution of patients, insufficient clinical trials is also an obstacle for the development of new therapies. Developments in translational research have a vital role in R&D research, and it requires samples with good quality in sufficient numbers. Biobanks meet these needs of translational research by providing good quality samples. Biological samples are very valuable and are needed for the sustainability of translational research. By creating a collection of high-quality samples and associated data, biobanks help to overcome the rarity of these samples. By this way, they provide opportunity to conduct clinical trials with low sample size. Biobanks collect biological samples and related data, store them according to ethical and legal guidelines, and provide quality assurance for each biobanking steps. To facilitate translational research, a new partnership is required between patients, clinicians, researchers, and industry. Biobanks potentially lie at the center of this relationship. Human biobanks expected as a vital component of translational research and helps to translation of basic laboratory research into human trials and translation of successful trials into clinical practice.
Before we get into details, I'd like to go over some definitions. We can define a biobank as a functional unit for facilitating and improving research by storing biospecimen and associated data. A biospecimen could be any sample which is taken from a living organism. It could be blood, urine, tissue samples, cells, or any biomolecules obtained from these samples. In this context, we can define biobanking as a process of storing biospecimen and associated data for future use. So, biobanks store human biological samples linked to relevant personal and health information for biomedical research. In this context, I'd like to emphasize that quality of samples and data and obtaining them under ethical norms are two critical issues for biobanking. Biobanks store samples and data according to international guidelines and international data protection regulations, as well as universal ethical principles. Classification of biobanks is not simple and can be made differently according to their mission. Scale, contents, design, stored tissue type, and governance structure can be used for classification of biobanks. They can be established for research or therapeutic purposes. According to Pan-European Biobanking and Biomolecular Resources Research Infrastructure, BBMRI ERIC, we can classify biobanks as population-based biobanks and disease-oriented or hospital-based biobanks. Biobanks need strong relationships with diverse stakeholders, including donors, clinicians, researchers, research institutions, regulatory bodies, and industry. These stakeholders have a strong influence on the organization, operation, and strategic decision-making processes of biobanks. The ability of a biobank network to disseminate biological samples and data depends on cooperation among its stakeholders. That's why networking is one of the key activities of biobanks to increase their impact. Storing samples and data of healthy people and patients is crucial for enhancing community health. Biobanking of high-quality samples and data facilitates the development of novel and effective prevention and treatment modalities, as well as the discovery of new diagnostic and prognostic biomarkers. It is known that availability of large biospecimen collections and corresponding well-documented, up-to-date epidemiological, clinical, and biological information is essential for translational research. In this context, biobanks provide a sustainable supply of good quality biospecimens to researchers, thus reducing the experimental bias and adding to the value of clinical research results. In addition, the outcome of these studies gives more opportunities for personalized medicine. In addition, biobanks create possible avenues to use big data better in translational medicine by bridging the gap between basic research and clinical applications. They can facilitate the translation of laboratory insight into clinical practice and industry. Biobanks can also help to create an infrastructure of data for translational innovation. In summary, biobanks accelerate basic and clinical research, shorten the path from research to innovative products, support the discovery of new diagnostics and therapeutics, and provide important economic impact. Rare diseases biobanks play a pivotal role by maximizing access to precious samples between researchers in the international scientific community. Registries of biobanks offer a unique, practical, cost-effective, and impactful solutions for rare disease research. They provide the appropriate resources required for the effective translation of basic research into clinical practice, contributes early diagnosis of rare diseases, discovery of new rare diseases genes, and development of new therapeutics. Biobanks are professional repositories of biological samples. They store biospecimens harvested over time combined with related dataset. Moreover, collected samples are processed, analyzed, and preserved in the biobanks according to standard operating procedures to share with the broader scientific community. Biobanks also provide detailed information about each sample's acquisition, handling, and storage steps, such as ischemia time, transport and storage conditions, and processing methods. Operational procedures for each biobanking activity must be established to prioritize privacy and autonomy, sample quality assurance for high quality, data privacy and protection.
Like all other biomedical research, biobanking also needs a good governance systems which covers the ethical, legal, and social issues. Now, when we talk about ethical principles, as we all know, there are very basic 4 types of principles, especially when we use human samples. One is the human dignity, benefit no harm, autonomy of the subjects, and privacy and confidentiality. Ethical principles must be safeguarded in each step of biobanking. Those are sample acquisition, sample process, sample and data storage, and sharing. Institutional review boards review the applications to set up biobanks, and they are responsible to monitor the ELSI principles that are in place. In sample acquisition step, obtaining an informed consent from donors is an essential process. In informed consent, donors must be informed about the purpose of the sample donation and any future use of those samples. Data collected and stored should be specified. What type of samples and data sharing will be used should also be specified. There are different types of consent forms found in the literature. One type is the general consent, where donors agree for their samples to be used in any unspecified future research. Another type of consent is specific consent, which is given only for one particular research. But for biobanking, a broad consent approach is preferred, which allows samples and data to be used without reconsenting from the donor for a defined framework of future research. For rare disorders, parental consent is usually used because rare disorder patients are usually minors and they should be reconsented at adulthood, either 14 or 18 years of age. This is the recommended process. There are international collaborations and collaborations with industry, data deposition in international registries. These are challenges that must be addressed in the consent forms. When we come to the processing and storage of data, transparency of the system must be met. The clinicians and sample donors should be informed about the workflow of the biobank. Systems must be operational for security and data protection. Access to the infrastructure and data must be restricted for designated personnel only. Data that is collected should be processed according to FAIR principles, that is, findable, accessible, interoperable, and reusable. To protect the autonomy and privacy of the samples and data, there are two types of coding that can be used. One is anonymization. Here, information does not relate to any identified person or to personal data. In other words, subject is no longer identifiable. Is this suitable for biobanking? Is a question. We'd rather use pseudo-anonymization, where subject can be identified indirectly or with additional steps. Because with research done on the samples, right to return of results is an ongoing ethical question. If you completely anonymize the samples, it becomes very difficult to inform the patients or donors about the results. Now, all this can be reviewed in data privacy guidelines. One very important document is EU General Data Protection Regulation that has been enacted in 2018. There are, of course, national laws and bylaws, but any type of work that is going to be done and any type of sharing of data with EU countries has to follow the GDPR guidelines. Sharing of samples and data for rare disorder translational research is important because the number of patients are restricted and may be geographically scattered. We need access committees for biobanks to review sample and data requests for further research, formulation of material transfer agreements for collaborative research contains clauses that define the rights of the recipient and the sample and data donor. Now, these type of policies and standards are shared by Global Alliance for Genomics and Health that can be used, and there is a good manuscript that is indicated at the end of the slide. For biobanks to have a good impact on translational research, they have to have a sound governance model. Biobank management has to have an accountability system. Awareness of biobanks and biobanking activities should be promoted in the general public and the healthcare system because trust of the population and all the stakeholders is a key factor. A plan for sustainability of the infrastructure and the sample and data should be designed so that biobanks can have a long-lasting effect on research infrastructures. Like all other biomedical research, biobanking also needs a good governance systems which covers the ethical, legal, and social issues. Now, when we talk about ethical principles, as we all know, there are very basic 4 types of principles, especially when we use human samples. One is the human dignity, benefit no harm, autonomy of the subjects, and privacy and confidentiality. Ethical principles must be safeguarded in each step of biobanking. Those are sample acquisition, sample process, sample and data storage, and sharing. Institutional review boards review the applications to set up biobanks, and they are responsible to monitor the ELSI principles that are in place.
Quality management system is an essential element of biospecimen management in the context of biobanks and biomolecular resources. It's a tool that includes quality assurance and quality control programs on biospecimen and biobank management. The key to implement an effective and sustainable QMS is the development and adherence to standard operating protocols. These standard operating protocols should address the all pre-analytical phase, include sample collection, processing, storage, and infrastructural requirements like security systems, computerized inventory, biospecimen quality tracking systems management, and equipment maintenance in the biobank. Here you can see the main components of quality management system. Quality management system should include quality assurance and quality control policies with written SOPs that describe pre-analytical phase, includes biological sample collection, processing, storage, and sharing of samples must be carried out. There should be standards and replicable protocols and SOPs for preclinical, clinical, and laboratory processes are critical for biobanks to ensure consistent operations. And biobanks ensure compliance with relevant biosecurity and biosafety requirements. The other component is data and sample management system. It should be a secure system where stored samples and data are continually matched with relevant health, clinical, and research data. And there should be a risk assessment that addresses risks and opportunities which keeps up to date. These all key elements are building blocks for any biobank to be successful and for translational research to thrive. In biobanking, it is critical to establish the relationship between clinical information and molecular knowledge, especially in the terms of personalized and translational treatment approach. ISO standards and guidelines have been developed for harmonization between various biobanks and networks. These associations and groups such as ISPER and OECD offer guidelines on developing a QMS with advice and templates available online for biobankers. In order to increase the value and quality of a biobank collection, they are excellent resources for those undertaking standardization and harmonization. Biobanks can also gain appropriate accreditation, thus improving trust from the research community. In order to achieve consistency in specimen management, some European biobanks are follow international ISO 9001 rules for quality management and more specifically ISO 2387 dedicated on biobanking. This standard addresses biobankers, researchers, and other parties for the applications on research and development. The basic intention of ISO 2387 promote confidence in biobanking, enable and demonstrate competent biobanking operations, provide biological material and associated data of appropriate quality, facilitates cooperation, fosters exchange, and assists in the harmonization of practices. According to this standard and guidelines, we can summarize QMS in biobanking under two headings: QA and QC of biological sample, QA and QC in the repository. Regarding the QA and QC of biological sample, the pre-analytical phase is crucial when handling specimen. Issues that arise even before samples arrive in the laboratory and further route to the biobank play an important role. Errors can occur during the pre-analytical phase with specimen handling and identification. Therefore, the pre-analytical phase must have strict quality assurance and control measures in place. State-of-the-art biobanking for complex or large trials represents a challenging task. Samples have to be collected and stored according to well-defined, rapid, and reliable processes. After storage, well-defined, rapid, and reliable processes have to be implemented as well, in order to guarantee high-quality samples as a compulsive basis for accurate analytical results in translational research. Furthermore, a series of details on the handling process, such as the time spent between sample collection and laboratory entry, centrifugation, or the time to freezing are documented as well. Biobanks should follow determined per-analytical standards to achieve successful interoperability of sample and associated data. The quality assurance and control of the biorepository is a combination of processes that interact with each other. Biorepository staff should be trained to adhere all relevant quality systems, SOPs, and quality handbook prepared under the guidance of ISO 23887 Biobanking standard. Internal and external audits should be carried out at specified time intervals, so the corrective and/or preventive actions should be taken. And a successful translational research program depends on the existence of biobanks with solid infrastructure that can provide the needed consistency in sample quality as well as facilitate disaster recovery plans and long-term financial stability.
Standardized processes for the collection, storage, and analysis of sample and data must be set for interoperability between biobanks. The predetermined procedures are very important for data and sample exchange among different biobanks. Standards must protect participants' privacy, confidentiality, and security with safety rules and laboratory practices prepared based on relevant law and regulations. Samples may require to be re-identified in limited circumstances under legal and ethical framework. It could only be done with prior permission of the participant and ethics committee. Data can be protected from unauthorized access with pseudo-anonymization. With this coding system, sample can be re-identified with additional steps by authorized people. Sample and data management should be done with transparent policies, protocols, and procedures specific for sample and data governance and sharing. Due to progress in biomedical research and omics technologies, metadata size related to samples are increasing. Many biobanks are using laboratory information management systems to support sample and data management and tracking. It is an electronic support of access procedures and includes sample history, quality, proper sample and data usage, and consents related. It also helps to manage sample, biobank workflow, storage, staff, and documents by real-time reporting systems. Standardized data managements help users to find which sample and data specific for their research purpose. For example, people are using BBMRI ERIC directory, the Biosample database, and RD Connect registry and Biobank Finder to navigate relevant sample and data. For proper data management and long-term sustainability of biobanks, data must be organized according to FAIR principles. FAIR principles has been developed to improve data findability, accessibility, interoperability, and reusability, which together provide harmonization, exchange, and sharing across biobanks. FAIR principles can be used for human sample and data, but it is also applicable to tools, algorithms, and workflows which create the data. To sum up, we can say human biobanks are the fundamental components of translational research. Translational research aims to strengthen and improve clinical practice and patient care. In translational pipeline, which defines the progressive parts of the research, biobanks have role in T1 and T2 research. T1 is a translation of laboratory research into human clinical trials, and T2 is a translation of successful clinical trials into clinical practice. The human biological samples become popular as a starting material, replacing the cell line and animal models. In addition, improvements in personalized medicine improves patient care. Therefore, human biobanks become a key element for all of those critical steps. Biobank sustainability is a vital concept for health research. It has three dimensions, which are financial, operational, and social. The resources available to biobanks are related to financial dimension. It must include development and management of a strategic plan. The quality and most effective use of resources are related to operational dimension. It should be based on best practices and standards. Also, it should have a good governance structure. The acceptability and trust in biobanks are related to social dimension. The acceptance of biobanking activities by public is very critical. I mean by patients, public institutions, governments, and funders. Therefore, transparent and fair access and sharing of samples are very important for gaining trust. Social dimension is also related to commitment to accepted standards. This creates a basis for measuring value. Some of criteria for measuring value of biobanks are presence of ethical review boards, membership to networks, and external quality assurance programs.